This poster represents a summary of a systematic review that was conducted to investigate the evidence of health-promoting effects of leaf teas on glucose metabolism. Leaf teas are widely purported as a treatment for many metabolic disturbances, including dysregulated glucose homeostasis. However, evidence on whether leaf teas can help to alleviate hyperglycemia or diabetes is scattered, and the putative mechanisms involved are not well documented. Therefore, our main aim was to evaluate the current clinical and cellular metabolic evidence supporting the use of leaf teas in the regulation of glucose homeostasis and identify the main tissue and cellular mechanisms involved. We conducted a systematic literature search of the recent clinical and animal nutritional intervention studies following the PRISMA guidelines. A total of 1,425 studies were identified, and after applying inclusion and exclusion criteria, a total of 97 studies were included. Of these studies, 14 were randomized control trials, 9 were population-based studies, and 74 studies were conducted in animal and cell culture models. The majority of studies retrieved focused on Camillus sensus-derived teas. Overall, they provided evidence that various types of teas may have potential beneficial and protective effects on glucose metabolism in humans, although not all studies agree. Of the 14 randomized control trials included, only 6 trials showed beneficial effects for glucose homeostasis, while 7 did not show any effects. Compared to the mixed results in human intervention trials, studies in animal models have demonstrated mostly positive results for the glucose regulation of leaf teas, with impacts in the pancreas, liver, muscle, and adipose tissue. A total of 50 animal studies showed improved glycemic control after tea consumption against the background of a variety of metabolic insults, including diet-induced obesity, while only 3 studies in animal models reported neutral or adverse effects after tea consumption. We noted 5 recurring mechanisms by which leaf tea consumption may promote insulin sensitivity, and they include the activation of insulin signaling pathway in the liver and skeletal muscle, insulin-independent glucose disposal in the liver, muscle, and adipose tissue, suppression of inflammation, and preserving liver function and protecting beta cells against oxidative stress. Several animal studies indicate that tea consumption may have beneficial effects in reducing diabetes risk. However, results from human clinical trials vary greatly and may be due to the heterogenic nature of the clinical trials methodology. Better designed clinical trials evaluating a wider variety of tea types are clearly needed to identify the benefits of tea consumption for human health.